Hello, this is Tober's Biology class, Chapter 12, Lesson 3. Tober's Biology class, Chapter 12, Lesson 3. At the end of Lesson number 2, we're talking about these special viruses that can make bacteria sick. They're called bacteriophages. And at the end of their cycle, they have many thousands of bacteriophages inside the bacteria until it bursts open. That is the end of the lytic cycle. Alfred Hershey and Martha Chase studied these bacteriophages, and they wanted to know what part of the bacteriophage actually goes into the bacteria. Was it the outside, the protein coat, or was it the DNA that was at the core? So here it says they wanted to determine which part of the virus, the protein coat or the DNA core, entered the bacteria cell. Their results would either support or disprove Avery's finding that the genes were made of DNA. And here's what they did. The outside, the bacteriophage, is able to be linked to a radioisotope, sulfur-35. And the inside, the DNA, they had to tag with phosphorus-32. These radioactive isotopes gave us the ability to look at the corresponding bacteria that came from this particular generation, and if it had phosphorus or sulfur, they could tell whether it was the outside or the inside of the bacteriophage that was injected. Since these proteins carried no phosphorus in the DNA and contains no sulfur, we use these radioactive substances to use markers to enable it. So in your notes, ladies and gentlemen, if you look at the bottom here, the bacteriophage with all the dots on the outside, that's the one that is marked with sulfur-35. And the one that has the dots on the inside is where the DNA is. So in the subsequent pictures or the pictures that are on tests and quizzes, you'll see bacteriophages either marked with dots on the outside or dots on the inside. And when you see that, it's based on these markers. The markers on the outside have to do with sulfur-35. The dots on the inside is phosphorus-32. So if they found the radioactive sulfur-35, that would mean the virus's protein coat went inside of the bacteria. Or if they found the phosphorus-32, they would know that it was actually the DNA core that had been injected into the bacteria. The two scientists mixed these marked viruses with bacterial cells and waited for a few minutes for these viruses to inject their genetic material. And then they tested that for radioactivity. Here's what they came up with. Nearly all of the reactivity of those bacteria was phosphorus-32. And that told them that it was the DNA that was injected into the bacteria, and then the subsequent generations after that was from the DNA being given off rather than the protein coat.
Hershey-Chase experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing P32 labeled deoxyribonucleotides. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
So, the role of DNA. What is the role of DNA in heredity? DNA is what makes up genes, capable of storing, copying, and transmitting genetic information with the cell. So, the analogy that we often give is DNA is kind of like a book that has been copied and passed around. It's got three basic functions: to store information, copy the information, and then to transmit that information. The foremost job of DNA, of course, is the storage of our genetic information. The genes control patterns of development, which means the instructions cause the single cell to develop to look at an oak tree or a sea urchin or a dog. So somehow it's all got to be copied and stored. So before a cell divides, it must make a complete copy of every one of its genes, similar to the way a book is copied in this particular picture. Many scientists was a puzzling aspect about how DNA has got to be copied, and it's got to be copied exactly, because if you leave a couple of the pages out, or if you leave information out, that could lead to different parts of the cells being replicated without the necessary information. Transmitting information. When a cell divides, each daughter cell must receive this complete copy of genetic information. If it's not complete, that can lead to all kinds of things like defects and things like that. So, if there's a loss of DNA during meiosis, that means that there might be a loss of genetic information from one generation on to the next. The structure of DNA. DNA is nucleic acid made up of what we call nucleotide, joined in long strands by covalent bonds. Nucleic acids and nucleotides 1. Nucleic acids are long. They're slightly acidic molecules, originally identified in the nuclei or the nucleuses of the cell. They're made up of what we call nucleotides that are linked together in long chains. What's a nucleotide? Here's what a nucleotide is. Nucleotide basically are these chains that are made of bases, deoxyribose, and a phosphate group. These are bonded together by covalent bonds. They come from these base pairs of A, G, C, T. The A stands for adenine, guanine, cytosine, and thymine. DNA's nucleotides are made up of those three base pairs. 5 carbon base called deoxyribose, phosphate group, and a nitrogenous base. Nucleotides in a strand of DNA are joined together by covalent bonds. So the covalent bonds are between the sugar and the phosphate group. So our base pairs, something that you'll need to memorize, and it'll be on the test, is what does the A stand for, the G, the C, and the T? Adenine, guanine, cytosine, and thymine. That's in DNA. In RNA, the thymine is changed for something called uracil. Nucleotides can be joined together in any order, so any order or arrangement of this A, T, C, and the G is possible. That is the end of lesson number 3.